I'd like to share the objectives and then the results from a multiple-year study that we conducted, and really at the outset want to express my appreciation to ILSI North America for really helping to allow us to study the prevalence of human enteric pathogens on melons that are produced and shipped from California and Arizona, with a very heavy emphasis on cantaloupe. And this study really came about through multiple discussions with our industry and industry produce associations and others in the wake of the very tragic outbreak of Listeria monocytogenes on cantaloupe that was attributed to a single grower and shipper in Colorado. And during that time frame, before many of the facts were known, a lot of people were projecting and predicting a level of risk and a level of concern for consumers that, in our experience, far outstripped and exaggerated the actual risk potential and risk exposure from earlier surveys that we had done, again, on behalf of the industry within California. We started with a preliminary baseline study funded by the California melon industry, by the Center for Produce Safety, and with certain cooperators that were very concerned about the economic damage that happened each time there is a large outbreak, or any outbreak, and a large recall of melons from outside of California. And I believe that work, I hope, was very instrumental in sharing the preliminary data with the reviewers at ILSI North America that ultimately awarded us a grant. And during that multiple-year process, before we started the project, we invited collaborators from the University of Arizona to join us, because an awful lot of the seasonal cantaloupe production and netted melon production originates from Arizona, and quite often it's the same growers and shippers. And so the few points that were very important to me in organizing the proposal then to be reviewed by ILSI were that we would be looking for the two primary pathogens of concern, Salmonella enterica and Listeria monocytogenes, on each melon, rather than splitting any lot into ones that would be assayed only for Salmonella, ones that would be assayed only for Listeria. Why that's important is it then dictates the methodology that we would use at the different stages, from a non-selective pre-enrichment of the whole melon, and then moving that into a selective enrichment to go one branch to Salmonella, the other branch to Listeria. So what we found is that within a population of melons collected at shipping point, so after harvest, after sorting, grading, packaging into cartons for shipping, that was the point at which we collected them. So what we say is that we tested melons from each of the targeted lots after all of the touch points had been accomplished. And for 98% or so of California melons and a large percentage of Arizona melons, that is the last point of human contact. So they go into the carton, they are what's called forced air cooled, so it's all air cooling, they're not washed, they're not immersed in a water bath, and they're not treated with an antimicrobial. And the first time that they are handled again would be at the point of purchase or the point of display at a retail market, or at the point of food service preparation or home preparation by the consumer. So we felt that in that way we could evaluate the risk of detectable levels of contamination on melons that were derived from the production areas that represent better than 65% of all the domestic source of cantaloupes, and of probably more than that of honeydews, and a smaller percent of the total watermelon production. So we focused largely on cantaloupe. So included in the fruit assays, we also spent some of our resources, some of our effort looking at environmental exposure, so looking for Salmonella, looking for actually the enterotoxigenic E. coli and Listeria monocytogenes and Listeria species, the indicator, in the production environment and within the farmscape. We did not detect either of the two primary pathogens, Salmonella or Listeria monocytogenes, in the first phase in about 4,000 melons, and then within the ILSI North America study, about another equal amount. We were not capable, or we did not find detectable levels in any of those fruit. In California, we did find 3 samples out of about 900 that, again, as composites, that were positive by molecular techniques and culture confirmed for diversity of serotypes, not a wide diversity, but a couple of different ones. So, low detection levels, we probably did 3 times as many as Arizona. So that might give us some indication of some differences in risk factors of contamination, within certain regions and seasons, because they're different, and other types of practices that might benefit from some additional study. So overall, I think we concluded once again that the risk of contamination of cantaloupes at the point that they leave the farm gate or the shipping point for distribution is extremely low. One thing I always like to emphasize is that people tend to project the outcomes of any of these research-based survey studies that are conducted really in one area to encompass everything. And we say this is for California conditions, California agroecology and practices, and may or may not reflect what risk profile you would have in a different area.